So I'm sending the request to record.
All right. So welcome to the NNE-CTR pilot project results final presentations for the spring of 2026. And so we have three talks today, and what we'll do is we'll just start off with the first one. Be Meagan Stabler, and she's Executive Director to NNE Co-op PCBRN, and her title of her presentation today is the Northern New England Provider Insights and Participatory Engagement NNE Pipe Survey. So Meagan, it's all on you now.
Great, thank you, Ron, for the introduction. Let me go ahead and share my screen, and folks, let me know if you can see it. Very good. Okay, thumbs up. Thank you, Sheila. Great. All right. Well, thank you for having me. I'm excited to be here and see some familiar, friendly faces in the crowd here. We're excited to talk about the NNE Pipe Survey. Before I dive in, I just need to give a little background about what the co-op is, because it provides really helpful context of this presentation. So the co-op is a primary care practice-based research network, or PBRN. We are the oldest PBRN in the country, started in 1972, and we currently have over 650 members and 150 practice member sites across Northern New England. We support clinician-driven research and quality improvement projects, and we work with a lot of different primary care practice setting types, so FQHCs, private practice, direct primary care, and of course, large academic medical centers like UVM Health Network. So that will just provide some context to this presentation. So outline, I'm keeping it pretty traditional: background, methods, results, and conclusions. So I shared what the co-op is, the Primary Care Research Network. We were inundated at the time of this proposal with survey requests. A lot of folks wanted, researchers as well as clinicians wanted to survey our clinical membership to tap into that rural primary care voice, which is a great thing to do. But not all of the proposals or the requests had compensation for the respondents, IRB approval, or also we were dealing with survey after survey with the same demographic questions, and it was just redundant. It was too much burden on our respondents, the co-op staff, and IRB. So that was our challenge. We needed a way to reduce the operational burden of the survey requests. And we were trying to think creatively of, could we reduce this burden at the same time as increase co-op interests and membership engagement? So our goal was to engage with primary care clinicians, generate data for their research endeavors as well as our operational endeavors, and to reduce overall operational burden. Before we came up with the solution, we looked in the literature, what's being done in this area successfully. And so we landed on the SURA survey, which occurs every year, and their approach, which West Virginia PBRN actually adopted about 10 years ago, is to request proposals, request survey questions from their members to then go back to the membership base and ask those same questions. So it's a well-practiced process. We just haven't done it in Northern New England yet in terms of primary care researchers. So we created the first of its kind NNE pipe survey to roll out among rural primary care providers in Northern New England. Again, our objective was to design a survey consolidation infrastructure that engages with primary care clinicians to generate data for their research endeavors while reducing respondent member co-op staff and IRB burden. And this objective aligns nicely with the NNE-CTR's mission to advance healthcare service research in rural areas, ensuring that the voices and concerns of those underserved populations are heard and addressed. So we solicited proposals of up to 10 survey questions, not including demographic questions. And we reviewed and selected 3 in a standardized way, which I'll show you that in a second. We received IRB exempt status from the DHIRB. We, after we collected all the survey questions, we built the REDCap survey, piloted it, and then disseminated it via the COOP and NNE-CTR listservs. We then made a clean, de-identified member level data that was, it was collected via snowball sampling, and therefore we didn't have a clear denominator value to be able to calculate a response rate. We had a drawing for respondents. Respondents could opt in to receive one of 15 $500 Visa gift cards. So just to recap, we provided the IRB exemption, one singular demographic questionnaire, and respondent compensation. So here's our timeline. We received funds from NNE-CTR July 1st, 2024, and we did a lot of work before that date in order to launch our new NNE Pipe Survey website on that day. So we launched our website to explain and accept proposals. Proposals were closed. The submission date was, due date was August 9th, 2024. We accepted 14 complete proposals that actually exceeded our expectation for this first go around, and all the proposals were really well developed. On September 9th, we selected 3 finalists and notified everyone of their proposal acceptance status. On October 1st, we launched the survey to the primary care clinicians. November 29th, that survey closed. And on December 6th, the 3 finalists of the proposals received their section of the questions as well as our demographic questions, which, as you can see, preceded the March 6th date of when that data became publicly available, aggregate, de-identified, clean. And then as a win-win here, all of the 3 proposal finalists were asked to come to the 45th annual co-op meeting to present their findings at the co-op meeting. So just to get a little bit more in the weeds of the how, the website creation, we created this logo here, which is the NNE Pipe survey. We leveraged our pre-existing co-op website, created a new tab on that website specifically for the pipe survey. That included the description of the survey, primary goals, eligibility criteria for the proposal submission, eligibility criteria for the respondents, the application process, instructions, FAQs, and we also included the proposal selection process and scoring criteria so the respondents knew what we were looking for before they drafted their proposal. We had 9 reviewers review all 14 proposals, unless they recused themselves from the review if they were involved with the application. So they scored on these following questions: benefits to rural primary care practices, establishes and enhances partnerships with co-op sites, focuses on social determinants of health and/or Northern New England predefined health priorities. Those are listed down here, and those were also on the website. These are from the 3 states China reports at the time. We also favored applications that we thought could result in data that would be helpful for pilot data on a grant or turned into peer-reviewed publications, accepted publications. And then generally just an overall high quality of submission. We had a handful of questions and we looked through the responses. So that was scored up top, and then down below, overall, would you recommend this to be included in our pipe survey? Yes, no. Key strengths, key weaknesses. And then if the reviewer wanted to include it, did you have any feedback to the PI on those questions? All 3 that were selected for inclusion, we provided minor feedback, and those recommendations were incorporated into the final questions. There we go. Okay, so we had a lot of outputs for this project, and we kept this very well documented because we would love to replicate this in the future. So we have a lot of templated emails, proposal advertisements. We kept those. We have acceptance materials, logo, website, text. It's archived now, but we still have it. Our survey built in REDCap and outreach materials. And then, of course, the reviewer forms, processes, and the IRB submission. Another output is increased co-op membership. So to submit a proposal, all of the lead author, the PI and the co-authors had to be co-op members in order to submit a proposal. We had 14 proposals received. We selected 3. So we had PCC and site demographics first, and then telehealth use patterns, primary care clinician role in discussing environmental risk, and this is really PFAS, radon, and arsenic. And then we had a climate-informed primary care climate health survey. And then at the end, we had just an opt-in opportunities for follow-up interviews, as well as compensation. People could opt in for compensation. In terms of some of our dissemination outputs from this, thus far, we've had 6 poster presentations from different presenters. You'll see it here on the right. These presentations were held at 3 different conferences, 2 of which were international. So it's really a launching pad for a lot of scholarship activity. We have 1 process paper that's under development currently, and then 2 content manuscripts that are being developed by the 2 of the proposal PIs, if you will. Yeah. So in terms of the results, I'm showing more of the demographics and engagement statistics. The content statistics related to the 3 surveys, the PIs are developing those materials for manuscripts now. So we had 173 PCCs complete the PIPE survey, and it took an average of 6 minutes and 7 seconds. We removed 28 responses because of duplication, missingness, or eligibility status. So the final set that is now aggregated, cleaned, and available is 145 unique PCCs across Northern New England. We had a diverse mix of practice experience type, and location. So you'll see here with the states, 35% were Maine, 30% Vermont, 32% New Hampshire. Doesn't total 100. We had a couple responses from upstate New York that were included in the UVM Health Network. Practice types, 45% were large academic medical center, but we also had a good mix of FQHCs, non-academic health system or hospital owned, private practice, and then 7 identified as other, and those others were direct primary care or community health centers. And 63% were female, which is actually lower than some of the CIRA big primary care surveys. So this is pretty well represented of what we can pull from national and regional information. 37% of the respondents had been practicing for over 20 years. And then the majority in family medicine who responded to this. And then in terms of bivariate association, again, with just demographics and some of the engagement questions, the only one that I thought was interesting to share is that there is evidence suggestive of an association between the state in which the respondent practices and requesting follow-up discussions on telehealth use at their practice. So you'll see Vermont was lower than New Hampshire and Maine. And I don't know why. I'm not sure if maybe folks on this call could highlight this for me, but maybe there aren't as many telehealth use issues in Vermont compared to Maine and New Hampshire. Not sure why they would be less interested in follow-up discussions on telehealth use. And then primary care clinician research interest, 142 out of the 145 respondents answered this question. This was fill in the blank. So that's, again, exceeded our expectation. This is really important information for the co-op operationally. I would say about half my job is being a matchmaker between clinician interest, practice site needs, and then developing research and resources, right? Funding to do the project. So this is one of the many ways that we collect information on what our members are interested in. So a lot of folks mention general quality improvement work in their practice, adolescent medicine, mental health, medical education. These 8 are more, I believe, like residency reproductive health, maternal and child health, diabetes, obesities, substance use, and health services research were themes that stuck out to us. So concluding, the PIPE survey was an innovative approach to engage rural primary care clinicians through participatory research that minimized respondent burden and enhanced scholarly outcomes and engagement within the co-op. So we found that structured data collection, selection, transparent dissemination helped foster this engagement and advance the academic productivity that we were looking for. All right, and we would love, again, I said lessons learned will inform future iterations of the PIPE survey. That's our goal. So please reach out to me if you have any questions. I'm happy to answer any questions that we have time for now. We have all the outputs and everything saved here, so I'm happy to share if folks are interested. And please feel free to become a member of the co-op.
Thank you, Megan. And so if anybody has any questions, pop up your hand or something so I can see, or just speak out loud. Any questions? Great. All right. Well, Megan, I think if someone has any questions, they'll get back to you. And so what we'll do is we'll move on to the next presentation, which is Benjamin King. He's Associate Professor at Bioinformatics at University of Maine. And he'll talk to us today about genetic factors of non-diabetic chronic kidney disease in Maine. And all right, Benjamin, it's all up to you.
Great. Hold on just a moment there. Yeah, so it's a pleasure to be here to tell you about my ongoing study looking at genetic factors of chronic kidney disease. And this is a study that we've been working on for several years now, and very fortunate to have help from Dr. John Vella as part of MaineHealth Nephrology, work with me and the team to expand our study using NNE-CTR pilot project.
Ben?
Right? Oh, yes.
We just see a blank white screen for your slide.
Hold on. Let me try sharing again. Thank you for telling me. Hold on.
That looks a lot better.
That's okay.
Perfect. Great. Thanks.
Great. Thank you for, the interruption. So, as I was saying, working with Dr. John Vella and as well as others at MaineHealth. And in parallel to that, have been working with Dr. Jim Jarvis at Eastern Maine Medical Center and his team that's part of the Clinical Research Center there to do a similar study in this part of Maine. And the overall goals of our study, having technology issues here. Screen. Hold on. It's not like I've never used Zoom before here. It's acting strange here. Hold on. Yes. Slide. There we are. Can you see my next slide?
Yes.
Yes, good. So we're interested in studying chronic kidney disease because normal kidney function is absolutely critical for maintaining good health. And with declining or impaired kidney function, increased risk for many other diseases like heart disease, and stroke, depression, and other diseases that can negatively impact the quality of life and Unfortunately, CKD is something that's a progressive disease and eventually results in kidney failure and is an important issue here in Maine, but of course across the US and also globally. It's estimated that 15% of adults have uh, chronic kidney disease. However, most are undiagnosed as there are no overt symptoms, especially early on in CKD. And there is a larger number of individuals who are at risk for developing chronic kidney disease. And globally, it's estimated that 10% of the population has CKD. And shown in this diagram, this is something that's important to Maine. And a study done in 2022, Maine had the fifth highest prevalence for CKD. Certainly, there are a lot of different risk factors for the disease. And so some of those risk factors include diabetes, hypertension, glomerulonephritis, which is inflammation in the glomeruli. Do you have different biological risk factors, environmental, and so on, as well as genetic risk factors. And so there are also interactions among all of those risk factors. So as CKD is this progressive disease, one clinical indicator is estimated glomerular filtration rate, or eGFR, that is used to classify individuals with these different stages, and I'll refer to these stages later as well. So as kidney function declines, eventually those individuals are on dialysis therapy with kidney failure. So to elucidate genetic risk factors, an approach used is genome-wide association studies, or GWAS. And what's done is to associate particular alleles of genes with individuals that have a disease versus those that do not. So looking at a case control kind of study where shown in this diagram, you'll have these single nucleotide polymorphisms, or SNPs, that are genotyped in individuals with the disease and those without the disease, and different technologies that are done to do that. In the past, a lot of those studies use microarrays to do those studies, where perhaps around 1 million SNPs are used to genotype the genome. And in this study, we're using whole genome shotgun sequencing. And so SNP number 1 is where there's not an association between a particular allele between the individuals with disease versus those without. The allele frequencies of those genetic markers are essentially equivalent. But the allele frequency within the population might be low, and that's why there are sort of a fewer number of these dots. You can also have SNPs that are more that have a higher overall frequency in the population, but again, there's no difference in associating them with cases versus controls. And then SNP number 3 is where there is a higher proportion of individuals that have disease and they have a particular allele. And so it's doing this association study, not just for 3 SNPs, but for millions of these SNPs. And so the primary objectives of our study was to identify putative genetic loci that increase risk for CKD, with some secondary objectives to look at associations between known risk loci among study participants, and compare the genetic data with other CKD studies as a way to assess whether study participants are representative of the cohort that we have. And so working with the team at MaineHealth Nephrology, as well as the fantastic folks that are there at MaineHealth Institute for Research. We worked to recruit patients, and then um, those individuals were screened and told about the study. And after receiving informed consent, then a blood sample was taken, and also clinical data was retrieved, as well as patient-provided information. The biospecimen and clinical data were de-identified, and the blood sample was shipped over to the MaineHealth Institute for Research, where it was archived until we had collected all of the samples, and then clinical data was coded into REDCap. Once we had the 32 samples, which was the goal for this particular pilot project, all of those samples were sent out at the same time. DNA were extracted, and then whole genome shotgun sequencing was performed. And then those results were downloaded here at the University of Maine for analysis. And then eventually we would be sharing the results through the NIH database of genotypes and phenotypes, which is a standard way of sharing these types of data to researchers that have appropriate credentials, and so that they can include the data in other similar studies. So the inclusion and exclusion criteria that we had are shown here. I won't take the time to go through all of them, but the important thing is that we were making sure to exclude individuals that had diabetes, as well as other forms of kidney disease, in the hopes of trying to find variants that we could associate with kidney disease that are not also contributing to these other diseases. And so we had 32 participant genomes characterized using whole genome shotgun sequencing, and we have had 65 additional participant genomes through the Northern Light EMMC study. And together with that, we're also leveraging data from the Alivus research program, where we've been analyzing a set of many larger cases, so over 22,000 cases, and a large number of controls in order to do the genome-wide association study analysis. So the purpose of this slide is just to show that the patients that we recruited met our inclusion and exclusion criteria, where we had individuals with stage 2 to stage 4 diagnosis, and then we have roughly equal number of male and female participants, and so this met our expectation. Then the work in terms of the analysis of the sequence data included taking the de-identified sequence data and aligning those reads to the reference genome assembly, and then calling sequence variants. And we used a Dragon workflow, which is a very robust way of aligning these hundreds and hundreds of millions of sequencing reads, and per individual, and then calling sequence variants. We also performed some secondary analysis of variants using a tool called eMedGene, where we're just taking particular SNPs and looking at their potential consequence, whether they are going to end up changing a codon that then changes the amino acid that's expressed in a particular protein, or the like, and whether that may contribute in some way to disease. So the process in terms of calling sequence variants ends up being where you have hopefully many reads that align a particular region that's shown here. So this is a small segment of chromosome 6. And what I'm trying to illustrate here is how there would be 31 reads that all could show a consistent mismatch with the reference. And this would be a homozygous sequence variant that's supported in a given genome with 31 reads. And that gives us confidence that with that number of reads that there is a sequence variant at that particular location. And then one can take those sequence variants and try to predict the consequence. Looking across all 97 genomes that we sequence, we have on average nearly 33-fold coverage, which means, like what I've illustrated here, there are 31 reads that cover this particular position. And so it would be 33 on average depth across the genome. Per individual, we had more than 4.86 million sequence variants. Of those sequence variants, nearly 4 million were SNPs, and again, per individual. And then we have other variants like structural variants, which are very long, more than 1,000 base pair segments that are different between individuals. And on average, we had over 10,000 structural variants. So with all of these, we can look to see what are the allele frequencies of these variants and try to associate them with disease. So one way that we've been analyzing the sequence variants is to take lists of sequence variants per individual labeled with these different case numbers. And these are ranked by their potential to alter the function of a particular gene. And so I'm just showing 6 or so variants just to illustrate this analysis. There is one consistent variant that ranks very highly per individual here, a variant on its complement 4B gene. And so the strategy here that we've been using is something called rank product analysis, where we're essentially taking the geometric mean of these ranks. Then we can use that rank product to list all of the variants and sort them in ascending order of that rank product and assign an overall rank. And so this complement 4B variant ranks very, very highly overall among all of the cases in this particular example. Another way to analyze these variants are to compare allele frequencies from our study as well as other studies of genetic variation, and one is the genome aggregation database, or genome AD, and compare allele frequencies basically with what we found in our study with other studies, and including this genome AD resource, and then look to see is there an association or not. And so these are just some of the sequence variants that come to the top of that list. And so we have some genes like mucin-1, which actually has been associated with another form of kidney disease, so autosomal dominant tuberointestinal kidney disease too. We've been using the All of Us research program data as a way to increase the number of genomes that we're analyzing, essentially. And so the All of Us research program has a goal to create one of the largest and most diverse databases of patient health record or electronic health record data, as well as whole genome shotgun sequence data. And so this is a fantastic resource that we're very fortunate to have. And so there are many individuals that have participated and have whole genome shotgun sequencing data. Some of those individuals have been diagnosed with chronic kidney disease, so we've been using those data. So with a genome-wide association analysis, that has been done with these data. It's been analyzing some 22,000 cases and using 267 controls. And some of the genes that come up, these are PKD1 are genes that are associated with autosomal dominant polycystic kidney disease. And so that's something that can be found using these data. And there are a number of other regions of the genome, though, that have been associated with risk of chronic kidney disease using these data. And one particular region is on chromosome 6 that I'm interested in. And so in that locus, which is quite large, it's nearly 2 million base pairs in length. There are 141 genes. And if one compares genes that we found through the allele frequency analysis, there are 6 that overlap. And then if we take the top 300 ranked variants, we find those same 6 genes. So there are 2 different complement 4 genes, complement 4A and B, as well as several MHC chains that come up in that list. Among the 27 that are shared between the allele frequency and ranked product analysis include PKD1 and mucin1 that I talked about earlier. So we continue to work on these with these data. And to summarize, the goal of the study was to find genetic loci that increase risk for CKD in individuals without major risk factors. And so we do have the 97 study participants, including the 32 from the working with the MaineHealth nephrology. And we've called sequence variants. We've been working on the secondary analysis through rank product and allele frequency analysis, and have been working with the All of Us data to do genome-wide association study analysis. I've presented on this work at a couple of meetings. Also tried to get some follow-on funding from the NIH through an R03 grant mechanism. Fortunately, that was unsuccessful. But future directions are to continue with the All of Us GWAS analysis, work on a manuscript, and then long-term try to functionally test these genes in zebrafish models of kidney disease. So the long-term goal really is to try to advance kidney research, improve patient outcomes, and using data like this, and to try to improve treatment. And certainly very much acknowledge all of the help from for this project, and especially the study participants. And included in that, of course, are the participants from the All of Us research program. But certainly appreciate all of John Vella's help, as well as other folks at MaineHealth and MHIR. So be happy to answer questions.
Great. Yeah, thanks, Ben. We have time for 1 or 2 quick questions. And so if anybody has any question,
yeah, actually, I was wondering, so you were showing the different variants ranked. You were saying there's differences between SNPs and structural variants. Were those ranked geometric differences also using structural variants within them? And also, like, what's the real difference, major differences between SNP variants and like structural variants in terms of like how the geometry of the system works together?
Great questions. So yeah, obviously SNPs, the S stands for single, so it's just a single base. But you do have haplotypes, right, across a region. So you can think of those individual SNPs being grouped in some way. And so different analysis methods do use haplotype information. The next category of variants beyond SNPs are what we call copy number variants, which are in the hundreds of base pairs. and there one could imagine a whole gene, perhaps. It's hard to imagine these things, but the essentially a region of the genome that's duplicated. And so a lot of the copy number variants, it's been widely used to in terms of cancer research and like diagnostics. There are copy number aberrations and things that are associated with some cancers. So there one could imagine maybe a whole gene. If there are more copies of that gene, then there's changes in the expression of that gene. It also could be a promoter region that's amplified essentially, and so that would then lead to changes of the expression of genes that are regulated by that promoter. Then yeah, structural variants are just larger scale, so in the thousands of base pairs. And so the analyses that I was showing with the rank product analysis, we do include all of the variant types with that. It's just the ones that float to the top of the list, because I couldn't show too many on the slide, those were SNPs. But the structural variants are there. What's hard is that because they're not widely studied, we don't have a lot of good data in order to look at essentially the allele frequencies of those structural variants. So thank you
Okay.
for the question.
The structural variants might be composed of like copies of sections of the genome, like SNPs that were just missense copies or something like that. But then they might have other
you
properties
can
that.
also there may not be any SNPs within that region that was amplified. And so it's not necessarily the case that they're always like loaded with SNPs or something. Yeah.
Thanks.
Yep. Great.
You have time for another question, one more question all right, quick, Ben, I have one quick question for you.
Sure,
So,
yeah, Rob.
looking at these complement factors that you found, like C4B, is there a role for autoimmune function in autoimmune disease in chronic kidney disease?
Yeah, I think there is an immune connection. Whether it's, autoimmunity or something, it's hard to know. But one thing that I've been looking at in parallel to this is looking out at how in different models of acute kidney injury in different animal models, there's a large, inflammatory response and things. And some recent work that Ian Drummond's lab is working on at NVIBL is narrowing in on some of the functions of neutrophils, in particular in different models of acute kidney injury. So, I think that yeah, this immune component is really important to that. Yep. Yeah.
All right. So so if you have any other questions for Ben, you can always drop them in the chat and contact Ben. So next speaker we have is Kahsi Pedersen. She's a faculty scientist at the MaineHealth Institute for Research. And Kahsi is going to talk to us today about assessing a mobile app-based mindfulness training to improve mental health of caregivers of children with autism spectrum disorder in rural areas. So take it away, Kahsi.
Thanks, Rob. Sheila, am I good to go to share my screen now?
I think Sheila said yes.
Okay.
Yes, I made you co-host, so you should be able to share your screen. Yay.
There you go. Looks good.
All right, great. Well, thank you for having me today. I'm going to present on my grant here. Oops. That was funded by the NNE-CTR. Again, yes, the study is titled Assessing a Mobile App-Based Mindfulness Training to improve mental health of caregivers of children with autism spectrum disorder who live in rural areas of Maine. So autism spectrum disorder is a neurocognitive disorder affecting about 1 in 38 American children by the age of 8, presenting with various challenges and of differing severity, such as impaired social interactions, restricted behavior patterns, and comorbid behavior problems such as aggression and self-injury. So caregivers of children with ASD experience much higher levels of stress, anxiety, and social isolation, leading to reduced quality of life compared to the general population and caregivers of children with other chronic health conditions, such as Down syndrome. This chronic stress and anxiety together are referred to as mental distress, which poses significant health risks, including physical inactivity, diabetes, suicide, drug addiction, and overdose. Maine ranks 7th in the country for ASD prevalence, and 61% of Maine residents live in rural areas. So we are one of the most rural states in the nation. And rural ASD caregivers, so caregivers of a child with ASD, they face additional barriers, including reduced access to diagnostic and treatment services, leading to delayed ASD diagnosis and higher emergency department use. These families are also at an elevated risk of adverse outcomes associated with both rurality and caring for a child with ASD. So it's really crucial to identify some scalable and accessible mental health resources that can effectively reduce stress and anxiety for these caregivers that are living in rural areas. So the problem we see here is that there is a high prevalence of mental health difficulties among caregivers of children with ASD living in rural areas of Maine because they face these unique stressors, including behavioral challenges, communication difficulties, and the need for constant vigilance. These stressors can, again, lead to increased levels of anxiety, depression, overall poor mental health, affecting both the well-being of the caregiver, their own personal relationships, and their ability to provide optimal care for their children. So mindfulness training, it teaches the practice of bringing awareness to cognitive, affective, and physiological experiences occurring in the present moment. It's been shown to be one of the most effective non-medication treatments for reducing stress and anxiety in multiple populations of caregivers, for example, cancer and dementia. And mindfulness training really promotes facing, and addressing unpleasant thoughts and sensations. It's highly effective in reducing stress, anxiety, and depression, and has shown positive outcomes for a variety of health conditions, such as anxiety, sleep, blood pressure, acute pain, substance abuse, post-traumatic stress disorder, cancer, weight loss, and smoking. But despite these promising results, much of the research has been preliminary and has lacked some scientific rigor. And more recently, it's been identified as beneficial for caregivers of children with ASD, but mostly in the sense of a mindfulness-based stress reduction class, which is in person and tends to be time and cost intensive. And so this poses additional challenges for caregivers in rural areas and caregivers of children with ASD needing constant care and vigilance. So accessible digital therapies such as app-based programs can assess these limitations and also provide vital support to rural caregivers within this population. So based on the literature that we've seen, we decided for this study to choose a mobile mindfulness intervention developed by one of my external mentors, Dr. Judson Brewer. He's a psychiatrist and neuroscientist and the director of research and innovation at Brown University Mindfulness Center. He studies the neural mechanisms of mindfulness and translating research into scalable behavior change programs, such as the one developed and used for this study, which was unwinding anxiety. Unwinding anxiety has been built on the clinical and mechanistic mindfulness training research. And this is a mobile app-based program which offers evidence-based mindfulness training with broad reach at a very low cost. It's a 30-day mindfulness-based application-based therapeutic program for stress and anxiety reduction. It delivers mindfulness training in a standardized, high-fidelity, and scalable manner, and has demonstrated anxiety reduction for up to 3 months post-training. Studies testing unwinding anxiety's efficacy have shown reductions in stress and anxiety for patients with generalized anxiety disorder, decreased physician burnout, and improved sleep disturbances. Other apps by the same developer have reported success in smoking cessation and reduced overeating. So while unwinding anxiety appears promising for stress and anxiety treatment, few apps have undergone empirical testing and validation, specifically within this population. And so I have just a little link here to share with you, which I'll probably have to do this. Can you see this now? Can you see this website now?
Yeah, we do. Yeah.
Okay, thank you. So this is the Unwinding Anxiety website, and it's just, it's nice. It shares with you, it's proven to reduce anxiety in the general population by 67% and to reduce worry-related sleep disturbances by 27%. And the app is designed to be just 10 minutes a day. So I'll just give you a little preview. There's just some music. So it's just something that you would kind of read through to get a sense of what this app is. And I'd like to share one more thing with you from this website here, which is just unwindinganxiety.com. It describes all the different pieces of the app for anyone who's interested in learning more about the intervention itself. We don't have a lot of time here to go through more, but this is an interesting, just quick anecdote from someone who has used the app.
I used unwinding anxiety because I was suffering from crippling panic attacks. Unwinding Anxiety is a program that uses evidence-based mindfulness practice to help us wake up to our habit loops causing anxiety, like worry thoughts of the future. And with consistency, non-judgment, and kindness, we can step out of those anxiety habit loops, hopefully forever.
So that's just an example of what the app is. Again, it's an evidence-based behavior change program aimed at reducing anxiety and worry-related sleep disturbances. You can do it for 10 minutes a day for about 30 days. So for this particular study, our research question was, does the Unwinding Anxiety app-based mindfulness training intervention lead to a reduction in stress and anxiety among rural dwelling caregivers of children with ASD? So specifically, will this population use this app? Will they find it feasible and acceptable? And is there any preliminary efficacy of the app at reducing stress and anxiety? Aim 1, to examine adherence and satisfaction with the app within this particular population of caregivers. And aim 2, to assess changes in caregiver stress and anxiety over the course of the mindfulness training. I'll walk you quickly through the methods. It's a clinical intervention trial pilot study. Our goal was to recruit 40 biological or non-biological caregivers. We were interested in 20 cohabiting couples residing together in a designated rural area of Maine based on the RUCA code. Inclusion criteria was 18 and older. They needed to be able to speak English, have a child with confirmed ASD diagnosis, have a smartphone, internet access, and willing to complete the online study assessments, the study intervention itself, and some follow-up assessments. We gave them, based on the recommendation of the app developers, we gave them 60 days to complete the 30-day intervention, so it wasn't adding stress and making them feel like they were forced to do something each day, and people found that to be very helpful. Participants were compensated, they were given the app for free, and they were also provided up to $200 based on their study completion. Unfortunately, this study was terminated prematurely, so we were able to collect a total of 14 participants, so 7 couples out of the 20 couples that we were hoping for. The average age of caregivers was 48, a very rural area here in Maine, so 100% white, 93 non-Hispanic. 71% with a mental health diagnosis, anxiety, chronic stress, or depression, a little bit more for mothers than fathers. And most of them were currently employed. About a third had completed a master's degree. And the average age of the child was 13 years old, and pretty consistent with ASD literature, significantly more males. Usually it's 80%. Our sample had 86, and they were diagnosed around the age of 8 years old. So with adherence, this is very interesting. We found that caregivers completed an average of 34 of the 30 modules. So we had some definite super users here. You're able to repeat any module you'd like after you do it. So the next module doesn't unlock until the next day. So you can't actually just binge all 30 modules in one day and just become super mindful overnight. So it is definitely a stepwise iterative process, but it definitely showed that many of the participants enjoyed specific modules and went back and rewatched them. Interestingly, male caregivers completed an average more than female caregivers, 42 compared to 29. The majority of the users found the mindfulness training intervention to be acceptable, satisfying, engaging, and effective, and they would recommend it to others. And the potential efficacy was 100% of caregivers felt that their anxiety justified using an intervention like this, and 98% of them found it useful for their own stress reduction. A few of the measures that we have here, I think one of the most important ones that we were looking at was the generalized anxiety disorder brief report scale to get a sense of how anxiety changed over time. And so we saw that there was a significant decrease in average anxiety scores over the trial from 7.6 to down to 4. And we also saw an increase in mindfulness in our sample. So we saw an increase of scores from about 35 to about 40. And the significant piece that we found for the mindfulness, which was really interesting, was I experienced moments of inner peace and ease, even when things get hectic and stressful. And for caregivers within this population, they have verbally expressed how hectic and stressful their daily lives are. So that's a really great outcome of this study, for sure. With the Pittsburgh Sleep Quality Index, the majority of our caregivers reported that they were above the clinical threshold for poor sleep, and which is consistent with the literature within this patient population. And then for child problem behaviors, we actually did see a decrease in their irritability scores, indicating tantrum-like or aggressive behavior over the course of the study, which is indicating kind of a spillover effect from parents increasing their mindfulness, decreasing their anxiety, and then we're seeing, a little bit of decrease in problem behaviors in their children. So definitely opportunities for future research there. And I'll just share that we have presented this work in several different areas over the past couple years. We presented this as the preliminary study as we were ramping up our study recruitment at the National Idea Symposium of Biomedical Research. I've had several summer students through the MaineHealth Institute for Research summer internship program. Several students have worked on this. We've had about four, we'll have another one this summer, and they have worked on this project to do their 3-minute talks and their poster presentations. So that word has definitely gotten out there through their work as well. This was also presented at a conference that I go to annually, which is the International Society for Autism Research. This was presented in Seattle and very well received, and also at the MaineHealth Institute for Research local research retreat here in Portland. And finally, this was presented most recently at the NNE-CTR annual co-op by one of the student interns, Lily Clifford. So next steps for this, we submitted, my co-principal investigator and I, Dr. Brianna Taylor from the Rue Institute, we submitted an R34 last June to the National Center for Complementary and Integrative Health. We're combining, she is a sleep researcher, and this is a very important field population who suffers from a lot of sleep risks, and we did find some interesting sleep data from this preliminary work. So we are going to resubmit our R34 on mindful rest, which is a community-informed feasibility trial of this unwinding anxiety mobile mindfulness intervention for caregivers of children with sleep-related safety risks. It was ranked pretty well, in the new middle third, and we'll be submitting that again this June. So this is just a quick snapshot of the aims we'll be looking at. It'll be community engagement studios to really look at the intervention and the active control conditions. And then we'll also be performing the pilot randomized control trial to test both intervention and control conditions. So I would like to thank all of our team. Myself and Brianna Taylor are the co-principal investigators, and we have a fantastic team with data managers, research assistants, again, the summer undergraduates. Of course, I'd like to thank the study participants for all of their work, the funders, NNE-CTR, and then also my mentors, Susan Santangelo, William Cook, Judson Brewer, and Matthew Siegel. So thank you for your attention, and I will stop for questions.
Thank you, Kahsi. So any questions for Kahsi?
I actually had one question. The drop from that you saw on the general anxiety disorder from like 7.44 to 4, does that indicate that those people are still on the general anxiety spectrum, or are they no longer after using this? Yeah, classified that way.
Yes, that's a great question. So whether or not that's clinically, it's clinically significant based on our patient population. But I'm not sure if that would still classify them as being as having the anxiety diagnosis. That's a great question.
Yeah. Thanks.
Welcome.
The questions? Great. Well, if you have any questions for Kahsi, you know where to reach her. So we can, I'm sure she'll answer them. So, well, I think that concludes the presentations today.